So I just want to say to everyone, first of all, thank you so much for being here today. I reiterate Brian's gratitude for everyone that it took to make this possible. Speakers, supporters, PCOM, of course, IDSA, all of our wonderful sponsors, and of course our AlzPI planning team, and then the whole AlzPI team. It is an absolute privilege to be here, and honestly, it's also a little bit surreal. 9 years ago, I never would have imagined I'd be standing in a lecture hall giving a talk on microbes in the brain or describing myself in the way that I will. So my titles are fine. What I actually do, I call collaboration architecture. So I bring people together. And what I bring people together around is the study of how chronic infections relate to chronic diseases, with emphasis on the brain and the lungs. But in order to really understand how I came to be in this sort of strange description of what I do, I have to kind of tell my story. And it did begin 9 years ago with being very sick. In my early 30s, I had actually just run my first half marathon. My kids were 3 and 5, and suddenly my well-controlled chronic lifelong asthma became severe and refractory to the point where I couldn't sleep at night. I also, over the course of a year, just began to deteriorate. I developed joint pain, a cardiac arrhythmia, a bladder pain disorder, which was one of the most painful things I've ever felt, brain fog, and the final assault at this terrible year were symptoms that were consistent with MS. It was, a time in my life that I feel like I was robbed of a year. So when I see pictures like this, I think about how much I missed. So I have been fortunate enough to have a background after college. I studied business, not science, and then I worked for a big pharma company and then a biotech company. And during that time, I learned how to read and understand medical research, and I always loved science. So when I got really sick, I started accumulating lots and lots of specialists looking for answers. And I had, by the time I was, a year in, about a dozen specialists. Each specialist, what I noticed was they were focused on the one part of my body. So I would go in and I would say, hey, urologist, why bladder pain and my joints? I had this inflamed joint in my hand that was painful. Why my cardiac arrhythmia? Why that I stopped digesting my food properly? Why do I have neurologic symptoms? And everyone with a specific body part would say, look, I don't know, but let's talk about what I can give you for your bladder pain. So I noticed there was this lack of integration, right? And everything was siloed. So the picture you see here, so I've undergone at that time 4 chest x-rays, a chest CT scan. I've had in total 3 brain MRIs in my time since being sick. Countless other diagnostic tests, a VQ scan to rule out pulmonary emboli at the time because my breathing was so bad. And this photo was taken of me post-op, still woozy from anesthesia after a colonoscopy because I wasn't digesting properly. But I was fortunate enough to come across someone and something that effectively changed the rest of my life. So in my illness, I began to fear that if someone didn't identify the root cause of why I was systemically inflamed, that I was going to die. So I ended up doing a Google search that changed everything. I typed in interstitial cystitis and atypical pneumonia, 2 of my diagnoses during that time. And what popped up was my friend Chuck, Charles W. Stratton IV. He was the director of clinical microbiology for Vanderbilt for over 30 years. He was a board-certified internist as well as medical microbiologist and ID doctor. He actually wrote the board exam questions for ID and microbiology. He was a brilliant, brilliant man. So I came across a study he did with a urology colleague at Vanderbilt where they took urine from women with the condition I'd been diagnosed with, the interstitial cystitis, and tested it for an organism I'd never heard of, Chlamydia pneumoniae. So everyone knows typically chlamydia, the sexually transmitted infection, that's Chlamydia trachomatis. I didn't know about pneumonia. I had no idea that this really prevalent organism floated around the human population. What the study showed, while it was small, too small to be powered to draw any huge conclusions, was that this organism that was detected in over 80% of the women's urine was capable of driving chronic inflammation because it hangs out inside our cells. And even though it's a respiratory tract infection, it travels throughout the body and immune cells. And at this point, my mind was blown. So I reached out to the corresponding author on this paper, Charles W. Stratton, Chuck. Never thought I'd get an answer. I literally wrote out my whole medical history and thought, he's going to think I'm insane. And within 5 minutes, I had a response. He said, you know, get tested. And he sent chlamydia pneumoniae, IgG, IgM. And, you know, the rest is kind of history. He ended up becoming my doctor. He ended up becoming my mentor when I started Intracell Research Group 7 years ago, unsure of exactly how I would help other than by bringing people together. And then he became my friend. And by the time he passed away 2 years ago, he was a part of my family. And you truly will never forget someone that brings you a torch in darkness. This man was about humanism in medicine. He was someone who truly cared about his patients. And I like to think that I was unique, but I wasn't the only one. There were other people who he showed his love and his gift to that had MS that he successfully was able to place into remission by treating infections. So what I got diagnosed with ultimately, I did have chlamydia pneumoniae infection, evidenced by direct detection actually that he coordinated through one of the academic labs actually. It's not even something that most people have access to. I do have Lyme disease. So Lyme is a bacterial infection that can mimic a lot of other things, but I think it's more than a mimic. I think these things are drivers. So I made a trip 3 months after Chuck initially did a telephone consult with my primary care doctor, who was open to starting me on 2 antibiotics. My breathing immediately improved drastically. My condition improved. I will say that being treated for chronic infections is no walk in the park. There's a picture up there of me having a Herx reaction, Jarisch Herxheimer. If you don't know what it is, look it up. But when a bunch of microorganisms like Borrelia die at once, or chlamydia pneumoniae for that matter, a number of things happen inside the body and it sets off an inflammatory cascade. In very much the same way that patients being given chemotherapy may get sick before they get better, the same thing is true of Lyme patients. That photograph was taken of me and my husband and children outside the Ryman Auditorium the first time I went to fly and meet Chuck in person. Because after the 3 months of treatment, he indicated that my treatment likely needed to be longer. So that's the point at which he overtook my care. And I can honestly say that being heard is so huge for people like me. And I know there are other survivors out here in the audience that have been impacted by this, but gaslighting happens. You know, you go around to different doctors and still there's just a lack of education. And so that's one of our big goals here today, is to provide that insight. So what really blows my mind, this is to get into the science of it, but in a really basic way, we know that infections cause inflammation. Many chronic diseases have their roots in inflammation. You go to the National Library of Medicine and you look up inflammation, the first thing on the list is infection. But yet, why isn't it in the differential? So one of the big things that's hard with chronic diseases and infection is this notion of causality, right? So if something is associated with something, doesn't necessarily mean it's causing something. And I love that. I call this my heavy seagull picture. But in this case, you know, we can use a picture like this to dismantle the discussion around microbes and Alzheimer's and say, well, you don't know that they're causative. They could be associated. True. However, what if the picture was somebody standing with a sledgehammer making contact with the fence? Do you still feel the same way? So I'm going to make the argument that microbes have the capabilities. They have the ability to get where they need to be. They have the ability, even from afar, to induce powerful immune responses, such as things that we observe with gut microbiome changes, as well as SARS-CoV-2, the COVID pandemic, you know, where something may not necessarily have to get in the brain to impact it. So this is just some of the evidence or categories of evidence that bring us all together today. Why are we all here about microbes and Alzheimer's? Well, they're found in postmortem AD brain tissues, so people that died with the disease, disproportionately, and different types of microbes than people who are healthy, age-matched, sex-matched controls. You also have evidence suggesting from one of our colleagues' labs here today, Dr. Will Eimer, and the late Dr. Robert Moyer, and Rudy Tanzi, Nanda Kumar, and others, that amyloid beta, which is the, quote, toxic substance that shows up in the brains of patients with Alzheimer's, actually might be an immune response or an antimicrobial peptide. You have the fact that animals that are inoculated with infectious organisms will go on to develop disease pathology in many different animal models that have been demonstrated. You have epidemiological data showing that people who get infections, almost of any kind, have an increased propensity later on in life to go on to develop dementia. And then one thing that is incredibly powerful that we identified in our work is an entity called reversible dementia. Now, I say we identified, we pulled together cases of infectious dementias and demonstrated that there are more than 80 in the medical literature, probably more that we miss, just depending on how you query it in PubMed, of dementias that patients presented with various forms of dementia, Alzheimer's, vascular dementia, frontotemporal dementia, others, and then were tested for infection, usually by lumbar puncture. Someone checked their CSF, not using any fancy technology, just using things that are available in a hospital microbiology lab, and found evidence of active infection, and then treated them with an agent and discovered that they could be, in the majority of cases, symptoms drastically improved. Some people were actually completely normal on an MMSE. That's compelling. And then we have the fact that there are other neurologic and psychiatric diseases that have been associated with or known to be caused by infectious agents. PANS and PANDAS that affect children, right? Neurologic conditions that are a response to strep infections. You have infectious neuroimmune issues across the human lifespan. So we have a new way of doing things here at AlzPI. We are really shaking things up by building diverse teams. It's a new model. It's an idea that shatters the concept that people that are Alzheimer's or neuroscience researchers should work on Alzheimer's. We have a team that is comprised of people like myself. I'm a patient. I am not formally trained in science. We have numerous specialties, and we encourage having debates, having meaningful discussions to make the most of what we can do together. And I'm so thankful to have a supportive spouse, my husband, Brendan, who's willing to creep around with me on construction sites to take pictures near silos. Thank you, Brendan. He's on Zoom. So, you know, like all things, they sometimes start out like a seed, you know, and then you grow them bigger. So that's the chicken scratch drawing of AlzPI, which was actually inspired by work by Dr. Michael Wilson at UCSF when I was reading about chronic meningitis and how we actually assess that. Dr. Wilson had a really cool small study where he tested patients with chronic meningitis, their CSF, using metagenomic next-gen sequencing, determined what microbial infections were there, found a diverse array of them actually, fungal, bacterial, parasitic, polymicrobial, viral, and then treated with targeted treatment to see if the patients got better, and they did. So I thought, oh my goodness, could we do that with Alzheimer's? Why not, right? So our team is really diverse. You'll see we have Dr. Breshwert, who's here today, who's a veterinarian. We have people working in biostatistics, data architecture and analysis, people working on fungus, people working on bacteria. We're intentionally diverse. And it turns out that this is not a bad idea. So this book is amazing. If you haven't read it, I highly recommend. But David Epstein asserts in a chapter called The Outsider Advantage that big innovation often happens from people that are educated, but they may be a little bit further from the problem such that they're able to reframe it. And in the book, he gives an incredible example of a situation at Eli Lilly with a gentleman named Alf Bingham, who started something, was basically like an innovation open source platform. And Lilly had these issues with some of their molecules that they were trying to resolve with this incredible internal team of chemists. And they cite this example where a patent attorney wrote into the open source platform and said, I have an idea based upon a tear gas patent I worked on. So literally solves the issue as an outsider. So this is what we're really trying to channel with our collaboration. And, you know, I have to say, the first thing we identified that was a major barrier to this space was the testing. So it was actually Dr. George Perry, who will be here later today, who pointed out, Nikki, how are you going to do this testing people and treating them, saying when the testing itself is inconsistent, right? We've seen studies come out on Alzheimer's and microbes. One lab finds something, one lab doesn't. Well, they're not using the same methods. There's no consensus. There's no collaboration. So we put together what we call this research roadmap, this establishment of a consensus protocol last year. It was published in Alzheimer's and Dementia, the Journal of the Alzheimer's Association. And for me, Coming from where I've come from as a patient to co-lead that effort with Dr. Richard Lathe and my amazing colleagues that are mostly all here today, was a major celebration in the Schultek house. That was the day our paper got accepted. And I pulled out one of the reviewer's comments, which is hopefully this paper will catalyze the collaboration for finding a cure for this disastrous disease. One reviewer referred to it as transformative. And I'm really happy to say this is actually a real paper on PubMed. Take a hot second to read that. We think the considerable physical distance separating our two universities, which prevented us from killing each other over minor details during the writing. So I will say everyone is marked safe after making the roadmap. You know, this huge team of scientists that came together. We did have some heated discussions. We wanted them, right? We want to debate things respectfully and disagree. You know, we can learn that way, right? We can avoid making mistakes. Everyone's healthy and safe. So this is a part of our paper in Alzheimer's and dementia that I'm super passionate about. And it has to do with reversible dementias that I mentioned earlier. 86 cases. Just like Michael Wilson's work at UCSF with chronic meningitis, very diverse microbes. This is just a quick snapshot of the top of the paper. I encourage you to go through the cases. They're pretty intriguing. Cryptococcus neoformans, Borrelia burgdorferi, various viral infections, polymicrobial infections. But what this really tells us is that it should inform research. Cases are considered in some ways by the community, the research community, from what I've heard, a low form of research, right? It's an N of one case. Well, that's one person. And I'm a person standing here today, right? And others are here today that are well because of treatment targeting microbes. And there's a lot that we need to understand, but it doesn't mean we should shy away from it just because it's complex. So what are we doing here? So our primary goal at AlzPI was 1, to publish the roadmap. That's been done. 2, multi-laboratory detection. So we've enlisted some of the experts in the world doing different types of detection methods on the same samples so that we can develop what we refer to as the consensus detection protocol. Once we have this protocol, we want to use it in living patients and see what we find. Do we find evidence of ongoing active infection? Do they respond to tailored treatment? Can we help people? How many are out there? Because right now, people aren't being screened for infections, and I think that's a problem. So what else are we doing? We're sharing samples. A sample of someone's brain, that's a life. We should be utilizing it to its fullest extent. We shouldn't have it at one lab, you know, so that it can have narrow analysis with metabolomics and then write a paper on it. We should do multiple different methods, looking at the host response, characterizing those tissues, looking at microbes, looking at pathology, look at all of it on those same samples. We're going to do that. We're publishing high impact reviews to harness the wealth of data that's already in the literature. So our consensus paper was the first one, and our second one is in press and will be out soon in a special issue of the Journal of Infectious Diseases, all about how microbes invade the sensory system and get in and also lead to sensory deficits and damage. We also are inspiring younger scientists and welcoming them into our midst. This is not just a whole group of people that's the same. We have junior people, we have senior people, and we want to create opportunities for younger folks to be both mentored and learn and grow within AlzPI. And then finally, education and awareness, things like we're doing today. None of this at all is possible, and I'm actually impossible for me to be here today without some of the people in these pictures and some of the people sitting in this room. Because literally, the research that's done on microbes and infection, even if it doesn't make its way into a guideline somewhere, you know, and change practice behavior on a broad scale yet, doctors like Chuck Stratton, who treated me, he was harnessing the literature. He was saying, Dr. David Hahn, your work on asthma, that's going to inform me putting Nikki Schultek on azithromycin for her asthma, right? So the work means something. It means something to people like me. And this team has been incredible. And unfortunately, we don't see each other that often in the flesh. It's mostly on Zoom. Some of us for many years, like Lavinia and I in the back row, worked together for 5 years before I saw her in person because she's in Switzerland. But this whole thing is possible because of our incredible team. And there are kind of a lot of us, and people are working with us in various capacities. Some people are sharing samples, data. Some are actually going to be study sites. Some people are going to just publish reviews with us, and others are going to help in other capacities, and we're always open to more collaborators. And finally, and definitely not, last but not least, I guess is the way to put it, we are so grateful for the funding support from these three organizations, and especially to the Sim Einstein Research Foundation Incorporated. Steve and Marilyn, your support has been incredibly unbelievable and generous, and it is giving us a chance to generate data to help us help people. And the same thing goes for the Godsnell Foundation and to All's Out. Thank you, Chad. I know you're back there today. Thank you, Kate, on Zoom. We're so appreciative.